every process of our body is designed intricately. We've seen many twists and turns in the journey of the cell cycle. We started the journey from the preparatory phase, that is the interphase, to finally reach our destination, that is the dividing phase. Can you tell me the name of the dividing phase? That's right. The dividing phase of the cell cycle that we have seen is called mitosis. During this informative tour, we've understood the complicated process of the cell cycle effortlessly. We hope the lucid ideas and the concepts were crystal clear. We've halted at many phases of the cell cycle. Can you name them for me? That's right. The G1, S and the G2 phases were included in the interphase. Prophase, Metaphase, Anaphase and Telophase were the phases of the karyokinesis, a part of the dividing M phase. This is then followed by cytokinesis, which results in the division of the cytoplasm. Let's have a quick revision of all these phases. The cell is fully grown and has replicated DNA and duplicated centrosomes while entering mitosis. We know this by now. So do you remember where does this duplication take place? That's the right answer. The genetic material and the centrosomes duplicate in the interphase or the S phase to be precise. And what about the other two phases of the interphase? Needless to say, G1 and G2 are growth phases which help in the enlargement of the cell. Now this full-fledged grown cell is all set for division. It enters the first dividing phase of karyokinesis, that is the nuclear division, which is called the prophase. We are familiar with the events that the cell undergoes in this phase. The chromatin condenses to form the classical chromosome structure, having two sister chromatids. We very well know the chromatin is nothing but the DNA associated with the proteins. So condensation of this chromatin begins in the prophase. And what happens next? Correct. The centrosomes move apart and begin releasing mitotic spindles. The nuclear membrane and the nucleolus also start disappearing, which marks the breakdown of the nucleus. After going through these changes, the cell enters the metaphase. Can you tell me the most important change occurring during this phase? That's right. Formation of the equatorial plate. The chromosomes align at the equator, forming the unique metaphase plate. Along with this, the cell undergoes many other changes as well. The centrosomes reach the opposite poles. Also, the mitotic spindle attaches to the kinetic cores of the respective sister chromatids. All these changes prepare the cell to enter the next phase of the karyokinesis, that is, the anaphase. Do you remember that anaphase is the shortest and most rapidly occurring phase in the complete karyokinesis? Yes, in this phase, the sister chromatids separate and begin their migration to the opposite poles of the cell. The cell then enters the last phase of the karyokinesis. We know this phase well, it's called the telophase. Here, the cell, two nuclei with daughter chromosomes are formed in the single cell. And this is possible as the phase carries out all the tasks reverse of what happened in the prophase. The chromosomes decondense to gain the relaxed string-like chromatin structure. And are we left with something else too? That's right, the nuclear membrane. The nuclear membrane and the nucleolus, which disappeared during the prophase, now start reappearing. So now the cell is all set for the last phase of the mitosis, that is cytokinesis. The phase of cytokinesis divides the cytoplasm of the cell. There is a formation of a cleavage furrow in the cell membrane which extends up to the core. And this results in the formation of two daughter cells. These two daughter cells are genetically identical to each other. In other words, they have an equal number of chromosomes in the nucleus. With this, we come to an end with the concept of mitosis. to learn some more interesting content related to cell biology? Do not forget to subscribe to our channel. Happy learning!